Azlan Adnan, M.A. presents The Global Odyssey of the Coconut Palm, how Austronesian mariners, Indian Ocean traders and European empires carried Cocos nucifera around the world. Friday, the 24th of July, 2026. Origins in the Indo-Pacific. The coconut palm, Cocos nucifera, is among humanity's most widely dispersed and economically significant plants. Today it grows throughout the humid tropics, from the islands of the Pacific to the shores of the Caribbean, from East Africa to the coasts of South America. This extraordinary distribution was not the result of a single process, but rather the cumulative outcome of natural oceanic dispersal working alongside successive waves of human migration, maritime commerce and imperial expansion over several millennia. Modern genetic research has transformed our understanding of the coconut's origins and global spread. While coconuts are capable of floating across oceans, genetic evidence demonstrates that the overwhelming majority of their present-day distribution reflects deliberate transport by seafaring peoples rather than random ocean currents. Their worldwide journey mirrors the history of maritime civilization itself, linking Austronesian explorers, Indian Ocean merchants, Arab navigators, Portuguese caravels and Spanish galleons into one continuous story. The primary center of origin and early diversification for Cocos nucifera lies within the central Indo-Pacific, an expansive maritime region encompassing islands Southeast Asia and the adjoining archipelagos of Melanesia. The vast area stretching across present-day Indonesia, Malaysia, the Philippines, Papua New Guinea and neighboring island groups possesses the greatest level of genetic diversity found in coconuts anywhere on Earth. In evolutionary biology, such concentrated genetic variation serves as the definitive hallmark of a species' ancestral homeland, confirming that primitive coconut palms evolved and diversified across these tropical archipelagos long before human populations began actively harvesting and transporting them across the open ocean. The two great genetic lineages. Genetic studies and modern DNA fingerprinting have revealed that the global population of Cocos nucifera subsequently differentiated into two principal, highly distinct genetic lineages. The first of these, designated as the Pacific group, originated in island Southeast Asia and the Western Pacific Basin. These palms exhibit structural and physiological characteristics heavily shaped by millennia of human selection and cultivation. Pacific coconuts frequently produce rounded fruits with relatively thin husks, high liquid endosperm volume, and a wide variety of semi-dwarf and dwarf cultivars specifically selected for ease of harvesting near human settlements. The second major lineage, known as the Indo-Atlantic group, developed further west along the margins of the Indian Ocean, particularly surrounding the Indian subcontinent, southern India and Sri Lanka. Indo-Atlantic palms generally grow much taller, feature stouter trunks, and produce more elongated triangular fruits encased in exceptionally thick fibrous husks. These wild-type traits conferred superior buoyancy and structural durability, making them well-suited to surviving marine transport. This group became the dominant coconut lineage across the Indian Ocean and was later carried by European maritime empires into the Atlantic basin. The historical application of molecular phylogeography and DNA fingerprinting since the late 20th century completely revolutionized botanical science, overturning many 19th century assumptions that viewed all global coconut populations as a single uniform species spread purely by natural currents. Genetic mapping proved that despite their distinct evolutionary trajectories, these two primary lineages remain fully interfertile. Wherever historical trade routes brought the Pacific and Indo-Atlantic groups into geographical contact, extensive interbreeding occurred, creating unique hybrid populations that preserve a genetic record of human maritime contact. Natural oceanic dispersal before human intervention. Long before organized maritime trade or human migration developed, the coconut palm possessed a remarkable evolutionary architecture tailored specifically for coastal survival and natural transoceanic travel. The mature fruit of Cocos nucifera is a specialized drupe possessing a thick, fibrous outer husk filled with air spaces, known as the exocarp and mesocarp, which surrounds a hard, impermeable inner shell or endocarp. This buoyant husk encloses a large supply of liquid endosperm or coconut water, which provides a vital reserve of moisture and nutrients for the developing embryo. This unique anatomy enabled wild coconuts to drop from coastal trees, survive prolonged immersion in salt water for weeks or even months at a time, and wash ashore on distant, uninhabited beaches with their internal embryos intact and ready to germinate in sandy, saline soils. Ocean currents undoubtedly enabled primitive coconuts to colonize nearby islands and emerging coral atolls naturally across short to medium distances within the Indo-Pacific basin. However, while natural ocean currents account for localized botanical establishment, natural dispersal alone cannot explain the structured genetic geographical patterns, the survival of fragile cultivated varieties, or the sudden emergence of coconut populations across vast oceanic barriers like the Eastern Pacific or the wide expanse of the Atlantic. The decisive factor in transforming the coconut from a localized Indo-Pacific coastal tree into a global agricultural commodity was deliberate human agency. The Austronesian expansion, circa 3500 BCE. The earliest great transoceanic expansion of the coconut palm coincided with one of the most extraordinary migration events in human history, the Austronesian expansion. Beginning around 3000 BCE from Taiwan, Austronesian-speaking seafaring communities migrated southwards through the Philippines and into the vast archipelagos of island Southeast Asia. These mariners developed revolutionary maritime technologies, including complex double-hulled vessels, outrigger canoes, woven crab-claw sails, and sophisticated open-ocean navigation systems based on ocean swells, wind patterns, and star paths, enabling them to cross thousands of kilometers of open ocean centuries ahead of similar seafaring achievements elsewhere in the world. By approximately 2000 BCE, Austronesian navigators had established permanent settlements across maritime Southeast Asia. Between roughly 1600 and 500 BCE, a distinct archaeological culture known as the Lapita movement emerged in eastern Melanesia. The Lapita people pushed the boundary of human settlement rapidly eastward into remote Oceania, reaching Vanuatu, New Caledonia, Fiji, Samoa, and Tonga. Crucial to the survival of these voyagers was the Austronesian transport package, a carefully curated collection of domesticated plants and animals necessary to colonize barren or isolated island ecosystems. The coconut palm occupied a central position within this portable agricultural ecosystem. Early voyagers carefully selected seed nuts for maximum water content, thick flesh, and ease of germination, storing them safely in the holds of their outrigger vessels. The coconut provided an irreplaceable multi-purpose survival kit: sterile drinking water for long ocean voyages, high-calorie food, cooking oil, timber for structural building, palm leaves for thatched roofing, and fibrous husks that were processed into coir cordage, essential for lashing together the structural components of their ocean-going canoes. Every successful landfall across the vast Pacific expanse resulted in the immediate planting of coconut groves, permanently altering the ecology of Polynesia. The settlement of Madagascar and the Western Indian Ocean, circa 500 BCE AD 200. Having successfully expanded eastwards across the Pacific, Austronesian navigators embarked upon a second, equally astonishing directional migration westwards across the full breadth of the Indian Ocean. From around 500 BCE onwards, these master mariners navigated westward, utilizing seasonal trade winds to traverse more than 6,000 kilometers of open water, eventually reaching the giant island of Madagascar off the southeastern coast of Africa. Comparative linguistics, biological anthropology, and modern human genetic studies have confirmed that the initial settlement of Madagascar was accomplished primarily by Austronesian voyagers whose closest direct linguistic relatives reside in southern Borneo, within the basin of the Barito River. Sailing in large outrigger vessels, these ancient navigators carried their traditional agricultural package across the ocean, including dryland rice, bananas, taro, yams, and substantial quantities of seed coconuts. The coconuts introduced into Madagascar by these early Austronesian settlers carried unmistakable Pacific genetic signatures. By the beginning of the Common Era, between the 1st century BCE and the 2nd century CE, established populations of Pacific-type coconut palms were flourishing along the eastern shores of Madagascar and throughout the nearby Comoro Islands. These early introductions established the first major Pacific coconut populations within the Western Indian Ocean, marking one of the earliest long-distance, transoceanic transfers of a cultivated tree species in human history. Arab, Persian, and Indian Ocean trade, AD 60–1500. While Austronesian voyagers were establishing Pacific coconut varieties in the southwestern Indian Ocean, a parallel chapter in the history of the coconut was unfolding across the northern and western sectors of the Indian Ocean basin. Maritime commercial exchange connecting the Malabar coast of southern India, the Persian Gulf, the Arabian Peninsula, and the Horn of Africa had flourished for centuries prior to the Common Era. Early classical writers of the Mediterranean world were well aware of these flourishing Asian trade networks. The anonymous Greek merchant manual, known as the Periplus of the Erythraean Sea, compiled around AD 60, explicitly details an active maritime trade network linking ports in Roman Egypt and the Red Sea, with the vibrant commercial emporia of Western India and the East African coastline, where coconuts were already traded as valuable commodities. During the first millennium CE, the process of Indianization across Southeast Asia further reinforced these commercial and botanical links. Expanding Hindu-Buddhist trading kingdoms like Srivijaya and Majapahit established sophisticated maritime networks that integrated Southeast Asian coconut production directly with Indian Ocean trade, long before the arrival of Islamic networks. From the 7th century onwards, following the rise and rapid expansion of Islam, Arab, Persian and Gujarati merchants dramatically expanded their maritime reach throughout the Indian Ocean basin. Dhows propelled by lateen sails crisscrossed the Arabian Sea, establishing flourishing mercantile settlements along the Swahili coast of East Africa, spanning from present-day Somalia southwards through Kenya, Tanzania and the Zanzibar archipelago to northern Mozambique. These Western Asian traders regularly transported seed nuts of the local Indo-Atlantic coconut lineage directly from southern India and Sri Lanka to supply their newly established coastal enclaves. East Africa, a meeting place of two coconut worlds. By approximately AD 700, permanent Arab and Persian trading posts were well established in coastal settlements such as the Lamu archipelago on the northern Kenyan coast. Between the 8th and 11th centuries, as maritime commerce intensified, coconut cultivation expanded rapidly along the entire Swahili coast. By around AD 1000, extensive coconut groves were firmly established along the coastal plains of Kenya and Tanzania, providing oil, timber and fresh water to the international crews of passing merchant fleets. This historical overlap of maritime trade routes transformed East Africa and its offshore islands into a grand genetic crossroads. In places like Madagascar, the Comoro Islands, Zanzibar and the Swahili mainland, the Indo-Atlantic coconuts brought by Arab, Persian and Indian traders encountered and growing side by side with the ancient Pacific-type coconuts introduced centuries earlier by Austronesian mariners. The result was extensive, spontaneous hybridization between the two distinct genetic lines. Modern DNA analyses of coconut stands across Madagascar, the Comoros, and coastal East Africa clearly reveal complex, mixed ancestries containing genetic markers from both the Pacific and Indo-Atlantic groups. These East African hybrid groves represent some of the most genetically diverse coconut populations found anywhere outside the ancestral Indo-Pacific homeland, serving as a biological document of intersecting human migrations. The arrival of the Portuguese in the Indian Ocean, triggered by Vasco da Gama's landmark voyage around the Cape of Good Hope in 1497 to 1499, systematically reinforced Indo-Atlantic coconut cultivation throughout the region. Portuguese colonial settlements in Mozambique and Goa, followed in later centuries by Omani Arab control over Zanzibar and the East African coastline during the 17th and 18th centuries, aggressively expanded commercial coconut farming and coir production across the western Indian Ocean rim. West Africa, a Portuguese introduction, 1498 to 1600. The botanical history of West Africa presents a complete contrast to that of the eastern coast of the continent. Unlike East Africa, which had been integrated into Indian Ocean trade networks for millennia, there is no convincing archaeological, historical or genetic evidence to suggest that coconut palms were present along the Atlantic coastline of West Africa prior to the European Age of Discovery. The introduction of Cocos nucifera to West Africa was achieved entirely by Portuguese navigators who pioneered the direct sea routes around the southern tip of Africa during the late 15th century. Following Vasco da Gama's initial return from India in 1499, Portuguese fleets established a continuous maritime corridor connecting Lisbon with Mozambique, Goa and Cochin. Portuguese captains routinely loaded resilient, heavy-yielding Indo-Atlantic seed nuts in southern India and Sri Lanka to serve as vital freshwater supplies during their long homeward voyages across the Atlantic. By the early decades of the 16th century, Portuguese sailors were deliberately planting these Indian seed nuts at strategic maritime stopovers along the West African coast. The volcanic archipelago of Cape Verde, settled by the Portuguese in the 1460s, quickly developed into a crucial agricultural staging post and redistribution center for tropical plants moving between the Indian Ocean, West Africa and the Americas. From Cape Verde and the island of São Tomé, Portuguese traders systematically introduced Indo-Atlantic palms into coastal regions, stretching from modern Angola and the Congo, northwards through the Gulf of Guinea to Casamance and Senegal. Because these introductions were drawn exclusively from Portuguese posts in India and Sri Lanka, West African coconut populations belong entirely to the Indo-Atlantic genetic lineage, showing no trace of Pacific ancestry. While these palms thrived in the humid tropical climate of West Africa, large-scale commercial plantations were developed much later. Historical documentation records extensive plantation farming in places like Badagry in present-day Nigeria during the 19th century, with European missionary accounts noting vast, thriving coconut groves established along the West African lagoons by 1876. Brazil, the first coconut colony in the New World. Brazil was the first country in the Americas to receive coconuts on a substantial scale. Following Vasco da Gama's successful voyage around the Cape of Good Hope in 1497 to 1499, Portugal established a vast maritime empire linking Lisbon with Goa, Cochin, Ceylon, Malacca, and after 1500 Brazil. When Pedro Álvares Cabral reached Brazil in April 1500, Portuguese authorities soon recognized that the country's long tropical coastline possessed environmental conditions remarkably similar to those of India's Malabar coast. Coconut palms therefore became one of the earliest useful tropical crops intentionally transferred from Asia to the New World. By the first decades of the 16th century, Portuguese ships were carrying coconuts from Goa, Cochin and Sri Lanka across the South Atlantic, often stopping at Cape Verde and other West African possessions before continuing to Brazil. Historical accounts indicate that coconuts were already being cultivated in northeastern Brazil by the 1540s and 1550s, particularly in Pernambuco and Bahia. These introductions belonged almost entirely to the Indo-Atlantic genetic lineage, reflecting their origins in southern India and Sri Lanka. The palms thrived in Brazil's humid coastal climate and spread rapidly along the Atlantic seaboard. Over subsequent centuries, they became deeply integrated into Brazilian agriculture, cuisine, and coastal landscapes. Today, Brazil ranks among the world's largest coconut producers, with extensive plantations stretching from Maranhão and Ceará southwards to Espírito Santo. The Caribbean, from colonial experiment to plantation crop. The spread of coconuts throughout the Caribbean followed closely behind their establishment in Brazil. Unlike the Pacific coast of the Americas, there is no convincing archaeological or historical evidence that coconuts were present in the Caribbean before the arrival of Europeans. Spanish explorers reached the Caribbean in 1492, but coconuts were introduced only after regular transatlantic shipping had become established during the early 16th century. The Spanish transported coconuts from their possessions in the Philippines and, later, from colonies in the Americas, while the Portuguese contributed additional planting stock originating from India and Brazil. During the 16th and 17th centuries, coconut cultivation gradually spread throughout the Greater and Lesser Antilles, including Cuba, Hispaniola, Puerto Rico, Jamaica, and Trinidad. As British, French, Dutch, and Danish colonies expanded across the Caribbean during the 17th and 18th centuries, coconut palms became increasingly common around settlements, plantations, and coastal villages. Initially valued as a dependable source of fresh drinking water for ships, coconuts soon became an important food crop and source of oil, fiber, and timber. By the 19th century, commercial coconut plantations had developed in several Caribbean colonies, supplying copra and coconut oil to expanding European industries. Genetic analyses show that Caribbean coconuts overwhelmingly belong to the Indo-Atlantic lineage, closely resembling those found in coastal India, Sri Lanka, West Africa, and northeastern Brazil. This shared ancestry reflects the maritime roots of the Portuguese and Spanish empires rather than any pre-Columbian introduction. The Atlantic coast of North America. The expansion of the coconut palm into the subtropical margins of the Atlantic coast of North America represents the final northern frontier of the plant's distribution in the Western Hemisphere. The primary locus for this establishment was the Florida Peninsula, where climatic conditions along the southern coasts provided the warmth and humidity necessary for tropical palm survival. Similar to the Caribbean experience, there is no evidence of pre-Columbian coconut cultivation anywhere on the North American continent. The initial establishment of coconuts in Florida occurred during the Spanish colonial period, driven by maritime traffic passing through the Straits of Florida. Spanish vessels sailing from Caribbean ports like Havana frequently carried coconuts as living water stores, leading to occasional plantings around early coastal settlements such as St. Augustine and key military outposts. However, the widespread naturalization of coconuts along the Florida coastline was accelerated dramatically by a famous maritime accident in the late 19th century. In January 1878, the Spanish brigantine Providencia, bound from Havana to Barcelona with a cargo of over 20,000 coconuts, ran aground off the coast of present-day Palm Beach. Local residents salvaged the buoyant cargo and systematically planted thousands of washed-ashore seed nuts along the barrier islands and mainland shores. This massive planting event gave rise to the dense coconut groves that subsequently defined the landscape of South Florida, inspiring the naming of Palm Beach itself. Modern genetic testing confirms that Florida's established coconut populations belong predominantly to the Indo-Atlantic genetic lineage, directly matching the colonial populations of the Caribbean and West Africa. The Pacific coast of the Americas, a continuing historical debate. The presence of the coconut palm along the Pacific coast of Central and South America prior to modern global trade represents one of the most intriguing and fiercely contested questions in tropical historical botany and biogeography. When early Spanish explorers under Vasco Núñez de Balboa first reached the Pacific coast of the Isthmus of Panama in 1513, and when subsequent Spanish expeditions explored the Pacific shores of Costa Rica, Colombia, and Mexico, they encountered established groves of coconut palms growing along localized coastal stretches. The primary historical source for this observation comes from the Spanish chronicler Gonzalo Fernández de Oviedo y Valdés, who in his monumental natural history published in the 1520s and 1530s, detailed seeing dense groves of native coconut palms along the Pacific coast of Panama. Oviedo specifically noted that these palms were already well known to local indigenous populations, who utilized them for food and water. The central debate among botanists, anthropologists, and historians revolves around precisely how these coconuts arrived on the Pacific shores of the Americas. Genetic analyses conducted on old-line Pacific American coconuts reveal a surprising scientific reality. These palms belong unambiguously to the Pacific genetic lineage, displaying close affinities to coconut cultivars found in the Philippines and Western Oceania, rather than the Indo-Atlantic coconuts carried by the Portuguese across the Atlantic basin. This discovery has led to two main competing scientific theories. The pre-Columbian Trans-Pacific hypothesis proposes that ancient Austronesian or Polynesian mariners transported Pacific seed coconuts across the immense distance of the open eastern Pacific Ocean, successfully landing on the South or Central American coastline centuries before the arrival of Europeans. Proponents of this view argue that the eastern Pacific Ocean currents are exceptionally challenging for passive coconut drifting, making deliberate human transport the most plausible explanation. This theory receives indirect support from broader archaeological and genetic evidence demonstrating pre-Columbian contacts between Oceania and South America, including the confirmed transfer of the sweet potato, Ipomoea batatas, from South America into East Polynesia around AD 1000, as well as chicken bones with Polynesian genetic markers recovered from pre-Columbian sites in Chile. The Manila Galleon hypothesis offers a post-contact explanation centered on the historic Spanish trade routes. In 1565, the navigator Andrés de Urdaneta established the tornaviaje or return route across the Pacific Ocean, initiating the famous Manila Galleon trade. For 2.5 centuries, between 1565 and 1815, massive Spanish galleons made annual round-trip voyages connecting Manila in the Philippines with the port of Acapulco on the Pacific coast of Mexico. These vessels carried enormous quantities of Asian trade goods, plants, seeds, and supplies, including living coconut palms used for fresh drinking water and shipboard nourishment. Proponents of this hypothesis argue that coconut seedlings transported aboard these Manila galleons were planted around Acapulco and subsequently spread northwards and southwards along the Pacific coast of Mexico, Central America, and Colombia via Spanish colonial coastwise shipping. The extraordinary genetic similarity between Pacific American coconuts and Philippine cultivars is entirely consistent with the Manila galleon trade, yet it does not fully disprove the possibility of earlier, small-scale pre-Columbian introductions by Polynesian voyagers. Consequently, the precise timing and mechanism of the coconut's arrival on the Pacific shores of the Americas remains one of the most fascinating unresolved mysteries in historical botany. Human agency and the globalization of the coconut. When viewed across the full expanse of human history, the modern distribution of Cocos nucifera stands as a monument to human maritime mobility. While the biological traits of the coconut, its impermeable shell, fibrous protective husk, and internal store of nutrient-rich liquid, originally evolved to facilitate natural dispersal along island coastlines, these natural mechanisms were utterly incapable of establishing the palm across global oceanic barriers on their own. It was human agency that transformed a localized Indo-Pacific coastal tree into one of the most ubiquitous and vital economic plants in the tropical world. The globalization of the coconut occurred in 3 grand historical phases. First, the ancient Austronesian mariners navigating without metal tools or compasses carried Pacific-type coconuts eastward into the far reaches of remote Oceania and westward across 6,000 kilometers of open ocean to Madagascar, embedding the palm into the cultural and agricultural foundation of the Pacific and southwestern Indian Oceans. Second, Arab, Persian, and Indian Ocean traders integrated the coconut into the commercial networks of the northern Indian Ocean and East Africa, introducing Indo-Atlantic genetic lineages that intermingled with earlier Pacific introductions, creating vibrant hybrid populations across the Swahili coast. Third, the European Age of Discovery unleashed a massive reorganization of global plant geography. Portuguese caravels carried Indo-Atlantic coconuts from India around Africa to establish them in West Africa, Brazil, and the Atlantic Islands, while Spanish fleets spread them throughout the Caribbean and across the Pacific via the Manila galleons. In the 19th and 20th centuries, European industrial demand for copra, coconut oil, margarine, and industrial soap transformed traditional smallholder coconut groves into vast, highly organized plantation landscapes spanning East Africa, Southeast Asia, the Pacific, and the Americas. Conclusion: a living chronicle of maritime civilization. Today, the coconut palm is so thoroughly integrated into the visual identity, agriculture, folklore, and cuisine of tropical coastlines worldwide that local populations almost universally regard it as a native plant. Yet, as modern genetic research and historical inquiry have revealed, the global presence of Cocos nucifera is not an ancient, static feature of natural ecology, but rather a dynamic historical artifact woven over thousands of years of human travel. Every coconut grove swaying along the beaches of East Africa, Brazil, the Caribbean, or the remote atolls of the Pacific represents a living botanical chronicle of exploration, trade, migration, and imperial ambition. The two great genetic lineages of the palm, along with the diverse hybrid zones where they overlap, map out the precise sea routes charted by Austronesian outriggers, Arab dhows, Portuguese caravels, and Spanish galleons. Far from being a simple gift of nature floating aimlessly upon the ocean currents, the coconut palm was carried across the seas by successive generations of mariners who recognized its supreme value for human survival. In spreading this extraordinary tree to every tropical shore capable of supporting its growth, these ancient navigators forever linked the continents, leaving behind a living legacy that continues to tell the epic story of humanity's connection to the sea.
Azlan Adnan, MA presents The Coconut's Secret Journey, Origins and Genetics, Wednesday, 22 July 2026.
For generations, scientists believed they had pieced together the story of one of the world's most iconic tropical plants. Yet groundbreaking genetic evidence is rewriting that narrative. Ancient coconut DNA, analyzed through cutting-edge genomic techniques, now reveals an extraordinary saga stretching back thousands of years. A tale of survival against pounding waves and salt spray, human ingenuity on the high seas, and the quiet power of a single species to reshape landscapes, cuisines, and cultures across the globe. Imagine standing on a sun-drenched coastline in island Southeast Asia, where the air hums with the rustle of broad palm fronds and carries the sweet, earthy scent of fallen husks warming in the sand. From these vibrant shores to forgotten ancient trade routes etched across vast oceans, the coconut's true history proves far more complex and captivating than anyone once imagined. Where did coconuts really come from? How did their hard, buoyant fruits cross seemingly endless blue expanses to colonize distant shores? And what role did early civilizations play in their remarkable spread? Today, modern science, drawing on advanced genomics, archaeology, evolutionary biology, and linguistic clues, offers compelling answers that bridge deep time with the intimate rhythms of human adventure. Two ancient ocean basin lineages. For decades, the coconut, Cocos nucifera, seemed to possess a murky or scattered origin, its wild ancestors lost in the mists of prehistory, and its presence across so many tropical regions explained vaguely by buoyant fruits drifting lazily on ocean currents. DNA analyses of thousands of coconut accessions worldwide have brought clarity, identifying two major, highly distinct populations that tell a story of parallel journeys rather than a single, simple path. The Pacific group traces its roots to the lush islands and coasts of maritime Southeast Asia, particularly the rich biodiversity hotspots of the Philippines, Indonesia, and Malaysia. Here, amid the humid embrace of tropical rains and the rhythmic crash of waves on coral shores, early peoples recognized the coconut's potential and shaped it through careful selection. This group displays unmistakable signs of domestication, the emergence of compact dwarf varieties, self-pollinating traits that ensure reliable harvests even in isolation, and the rounded neovi fruits optimized for human use, with a higher ratio of sweet, milky endosperm to tough, fibrous husk. Crack one open under the blazing sun, and the cool, refreshing liquid inside offers instant relief, while the tender white flesh delivers a subtle sweetness that has sustained generations. Austronesian seafarers, master navigators of the Neolithic era, carried these cultivated coconuts, including prized forms such as the compact Fiji dwarf, Niuleka, on their epic migrations across the vast Pacific in outrigger canoes laden with the promise of future groves. Genetic echoes even reached the Pacific coasts of the Americas long before European contact, preserved in ancient DNA signatures and subtle archaeological traces of early cultivation. In contrast, the Indo-Atlantic group emerged along the southern shores of the Indian subcontinent, including the palm-fringed beaches of Sri Lanka and the Maldives, where the air is thick with the briny tang of the sea and the distant calls of seabirds. Cultivated independently by ancient Dravidian societies, these coconuts, often tall, cross-pollinating types with larger, more angular fruits, became staples of coastal life, their swaying silhouettes dotting the landscape as they spread westwards. While they generally lack the full suite of refined domestication traits present in their Pacific counterparts, the existence of these two separate centers of early cultivation highlights a fascinating pattern. Humans independently recognized and nurtured the coconut in different ocean basins. Yet the Pacific Southeast Asian lineage ultimately became the dominant force behind its global success, its fruits traveling farther and influencing more lives. These genetic revelations gain even greater depth when woven together with archaeological finds, ancient texts, and linguistic evidence. Coconuts did not merely float passively across the seas. They rode the waves of human history as vital companions. For Austronesian voyagers slicing through turquoise waters, the palm provided crisp drinking water that stayed fresh for weeks, nourishing flesh that could be eaten raw or dried into travel rations, and strong fibers twisted into ropes and sails that whispered reliability with every gust of wind. Trade routes carried Pacific coconuts to Madagascar and the East African coast, where genetic admixture with local Indo-Atlantic populations still echoes those ancient exchanges in the mixed groves swaying under monsoon skies. Later, Arab merchants gliding along spice-laden winds, followed by European colonizers and the engines of global commerce, blended populations further, planting coconut groves that now fringe tropical shores from the Caribbean's turquoise bays to the warm currents of the Indian Ocean. The architecture of dwarfism. Hormone mutations. The transition from tall, wild-type palms soaring over 20 to 30 meters high to compact, early-bearing dwarf trees represents one of the most significant triumphs of ancient Southeast Asian selection. Understanding how a giant palm was transformed into a manageable, highly productive food source requires peering directly into the molecular mechanics of the plant's growth hormones, specifically the gibberellin, GA, pathway. Gibberellins are diterpenoid hormones that drive cell division and stem elongation. In tall coconuts, uninhibited gibberellin synthesis leads to long internodes, delayed flowering, often requiring 6 to 10 years or more, and massive vertical height. In dwarf varieties, mutations within this pathway interrupt normal stem elongation, yielding short internodes, a compact canopy, and precocious flowering within just 2 to 4 years of planting. Genomic sequencing of tall and dwarf cultivars has isolated 2 major regulatory hubs responsible for this morphological change. 1. GA20-oxio-X, biosynthetic gene regulation. The enzyme GA20-oxidase catalyzes the rate-limiting steps in producing bioactive gibberellin. Rather than a total knockout mutation, which would render the palm sterile or severely stunted, dwarf coconuts possess a refined set of quantitative genetic modifications. Copy number variation, CNV. Tall coconut genomes carry greater copy numbers of key GA20-ox genes, typically up to 5 functional copies, compared with dwarf genomes, which frequently carry only 3. This reduced gene dosage directly lowers the overall volume of synthesized growth hormone. Promoter single nucleotide polymorphisms, SNPs. High-throughput sequencing reveals a crucial single nucleotide change in the promoter region upstream of the GA20-ox gene on chromosome 12, locus GZ to G04578. This variation significantly suppresses promoter activity in dwarf tissues, lowering gene expression by anywhere from 1.3-fold to more than 12-fold compared with tall palms. CNGA20-ox1 versus CNGA20-ox2. While coding sequences for CNGA20-ox1 are largely identical between tall and dwarf variants, expression levels of CNGA20-ox1 in leaf and stem tissues are substantially higher in tall palms. CNGA20-ox2 remains low or tissue-specific in both, indicating that regulation occurs primarily through promoter elements and gene dosage rather than structural protein disruption. 2. DELA signal repressors and growth regulators. Downstream of hormone synthesis lies the signaling mechanism governed by DELA proteins, such as CN-DELA1, and growth-regulating factors, such as CNGRF1. DELA proteins act as master negative regulators of growth. When gibberellin levels are high, DELA proteins are targeted for degradation, allowing the plant to grow tall. When gibberellin levels are low, DELA proteins accumulate and suppress growth. Targeted PacBio long-read sequencing across diverse coconut populations has revealed key genetic variants within CN-DELA1, including specific deletion patterns and diagnostic markers, such as MN-DELA1 score SNP1270 and MCN-GRF1 SCN672. These markers correlate strongly with the dwarf phenotype, modulating the palm's sensitivity to available gibberellins and reinforcing the compact growth habit. Together, reduced GA synthesis and altered DELA repressor dynamics create a balanced, highly productive dwarf tree. 2 distinct dwarf paradigms. As human migration and trade carried coconuts across different regions, selection pressures created distinct dwarf varieties suited to local environments and human needs. The Malayan dwarf of maritime Southeast Asia and the Chowgat green dwarf of southern India represent 2 of the most prominent, well-characterized dwarf varieties in modern agriculture. The Malayan dwarf, the Pacific paragon. Originating in the heart of the Pacific lineage in maritime Southeast Asia, the Malayan dwarf, occurring in distinct color forms, green, yellow, red, and golden, is a premier example of human-driven domestication. Driven by a mutation enabling strict autogamy, self-pollination due to overlapping male and female flowering phases, the Malayan dwarf achieved rapid genetic fixation. SSR marker studies show high levels of genetic uniformity within these color lines. The tree reaches a modest 4 to 10 meters, bearing vivid clusters of rounded, high endosperm fruits close to the ground. Aside from its early yield in sweet water, the Malayan dwarf gained global agricultural prominence because of its natural resistance to lethal yellowing disease, a phytoplasma disease that decimated tall coconut plantations across the Americas and the Caribbean. The Chowgat green dwarf, the Indian sentinel. Hailing from the Chavakad region of Kerala, India, the Chowgat green dwarf, CGD, alongside its orange variant, represents a parallel outcome within the Indo-Atlantic sphere. Reaching 4 to 5 meters in height and flowering within 3 to 4 years, CGD also exhibits nearly 100% self-pollination, resulting in remarkable phenotypic and genomic homogeneity across generations. Chromosome-level assemblies of the CGD genome, approximately 2.68 gigabytes, have revealed more than 300 resistance gene analogs, RGAs. Unlike the Malayan dwarf's reputation for lethal yellowing resistance, CGD was selected for its exceptional tolerance to root wilt disease, a devastating condition endemic to southern India. Its medium-sized, bright green nuts remain a staple for tender coconut consumption across the subcontinent. The co-evolution of humanity and the tree of life. Despite centuries of movement, interplanting, and commercial hybridization, the genetic fingerprint of those original Southeast Asian domestications remains strikingly visible in coconut populations worldwide. This persistence underscores a profound truth. While the coconut is a marvel of natural adaptation, its buoyant, salt-tolerant fruits perfectly suited to coastal life, enduring sun-scorched beaches and storm-tossed seas, it is also a deeply human plant, co-evolved with the peoples who cherished its shade, sustenance, and versatility. More than the biography of a single species, the coconut story offers a window into the intricate interaction between humanity and the natural world. From the coastlines of island Southeast Asia, where its domestication first flourished amid the perfume of frangipani and the distant roll of thunder, the coconut has reached tables, economies, and ecosystems around the globe. Split open on a market stall, its aroma evokes distant islands. Pressed into oil, it lights lamps and soothes skin. Woven into daily life, it supports entire communities. Today, chromosome-level reference genomes, transcriptomic analyses, and marker-assisted selection allow breeders to pair the compact stature and disease resistance of Pacific dwarfs with the rugged vigor of ancient tall lineages. Whether through the lens of evolutionary genomics, archaeological discovery, or the simple pleasure of tasting cool coconut water on a parched tongue, Cocos nucifera stands as a living testament to how human curiosity and plant genetics intertwined to shape the tropical world.